Hi, my name is Chloe Atkins, and I'm from the Bridgeport Regional Aquaculture Science and Technology Education Center. My project is on the non-thermal induction of heat shock protein 90 to mitigate early oncogenesis with preserved HSR. Already on the left here, you can see HSP90 itself and its close conformation, but you may be asking, what are heat shock proteins? Heat shock proteins, or HSPs, are a group of proteins which are produced during stress to protect cells in one of three ways. They work to refold proteins properly, prevent protein aggregation, and degrade dangerous proteins. They are part of the heat shock response, also known as the HSR. The HSR is an upregulation of HSPs during cellular stress. Stress could range in anything from a fever, severe injury, disease, and even cancer. There are numerous other environmental stressors as well, but it depends on the organism since HSPs are found in all living organisms. There are also many types of HSPs which are classified by their molecular weights. Two of the most common types are HSP70 and HSP90. So HSP70 constitutes of one half of all heat shock proteins. They fold nascent or baby proteins, which are proteins that are not already active, but they're going through their primary, secondary, tertiary, and quaternary structures. Also, HSP70 is thought to only be thermally activated or temperature activated. Um, Lastly, HSP70 delivers complex proteins to 90. HSP90 works on complex or already active and since denatured proteins. Proteins like an antibody that has since been denatured, and 90 works to recruit the protein and refold it back to its correct structure. It is also believed that 90 is activated by both thermal and non-thermal stressors. So in the bottom left, figure A, you can see HSP70 in its open conformation, and then figure B in its closed conformation. You can see there's a linker in between. It kind of acts like the clam to like hinge shut to lock the protein inside to create optimal folding conditions. Versus you have on the right over here, you have HSP90, figure A in its closed conformation, B in its open conformation, and the client binds to the middle domain. Um, like I said, HSP70 delivers complex proteins to HSP90. You can see the cycle demonstrated here where you have the HSP70 complex with HSP interacting protein and heat shock protein 40, which recruits the client and delivers it to the middle domain of HSP90, where you can see over here. And then once it delivers the client, you can see ATP entering, which causes HSP90 to snap shut and the HSP70 complex to leave, also taking HOP or HSP organizing protein with it. You then see ATP hydrolysis occurring because you have the byproduct of an inorganic phosphate. And then once the phosphate's released, you can see HSP90 opening into its open conformation, releasing the client. So this is when HSP90 is acting as a foldase and actively refolding proteins, but 90 and 70 can also act as a holdase, where they bind onto denatured proteins and they don't refold them, but they just prevent further aggregation. You can already see that there's many interactions occurring here. So in this picture here, you can see broader interactions of HSP 70s and 90s. So you see 70s in the middle, which if it's a nascent protein, it'll refold the protein to the structure it found it in. But if it's a complex, it has the option to deliver it to 90 to then refold it properly. And then HSP70s could also decide if the protein is too far denatured, it can send it to proteasomal degradation or lysosomal degradation. Another really important thing to note here is you have JNK and AIF, which HSP70s are activating. These are two stress-activated kinases, which then go and block apoptosis. Apoptosis was what would eradicate dangerous cells and things like cancer. So this is what makes HSPs directly implicated with cancer metastasis. So cancer is a stressor in itself. At some point from benign to malignant transformation, cancer activates the HSR. And once the HSR is activated, it aids cancer in two ways. One, it manages the influx of cellular byproducts, and two, it blocks natural eradication pathways, like I said, like apoptosis. It manages the influx of cellular byproducts since cancer is the dividing of cells without stopping, and it's producing more and more proteins that the body wouldn't normally be able to manage. So it relies on the heat shock proteins to kind of manage this excess protein production. Like I said, it's proven in mouse models that HSPs are needed for malignant transformation, and this was proven in 2014. So when HSPs were inhibited, malignant transformation did not occur. Because of this, HSP inhibition is a potential future to cancer therapy. But the downside to this is you're stripping the body of its natural processes, which would leave you susceptible to proteotoxicity. So if a patient was receiving an HSP inhibitor and they contracted a fever, they would not die from the cancer, but they would die from proteotoxicity. This is why some HSPs are needed to protect the body. This project is attempting to do just that. There's an attempt to only induce HSP90 with a non-thermal stressor. This would achieve a compromise between preserving the HSR and mitigating the spread of cancer. Mitigating the spread of cancer would be achieved by indirectly inhibiting the activation of HSP70s, which would enable apoptosis to function more. Also, the HSR would be preserved in some aspect since some 90s would be activated just in case as a safeguard in case further stressor did occur, like a fever. Since HSPs, like I said, are found in all organisms, Saccharomyces cerevisiae is used as my model organism, and also because HSPs interact with over 10% of the yeast proteasome. For my non-thermal stressor, I'll be using UVB bombardment. In theory, HSP70s will not be activated since it is non-thermal. If it is possible to selectively activate 90, it has a strong potential to glean more about the HSR. The HSR is still a very new and unknown area of study that is not mainly taught in textbooks or even many classes, and there's many gaps in this area. So this project could gain more knowledge and insight into can only 90 be activated, and if only 90 can be activated, can it even function independently of 70? Also, like I said, it has the potential to become a safer cancer treatment, kind of keeping some of your body's natural processes still in place, and it would mitigate the spread of cancer, like I said, because you have some HSPs as a safeguard, and you can see this demonstrated in the figure to the right. So you have UVB stress, which would activate HSP90, leading to protein protection, but you would have no activation of HSP70s, which would equal to less apoptosis inhibition, which would lead to cancer cell degradation. And you can see a direct, like 50-50 compromise between cancer cell degradation and protein protection. Also, this does have the potential to promote selective HSP inhibitors. So instead of inhibiting the heat shock response as a whole through inhibiting heat shock factors, it would be through only inhibiting one particular HSP. Like I said, for my project, I'll be using brewer's yeast, and so my procedure starts with inoculation of brewer's yeast, which is grown for 1 hour, and then a pre-stress cell count, which is taken using a spectrophotometer at 600 nanometers with a correlating cell count equation at 600 nanometers in a spectrophotometer. And then UVB stress occurs in a light-controlled box, and a post-stress cell count is taken, and then an electrophoresis is ran. And electrophoresis kind of denatures and unwinds proteins. So picture like a protein's like a piece of curly hair, picture it like pulled straight, and then it's pulled through the gel based on charges, which then, based on how far it travels, would correlate with what protein it is. So you'd expect 70 to be 70 long, 90 to be 90 long. And so, but before you can actually measure that, you need to activate the gel again with UVB light, and then you're able to do this molecular weight estimation. More into the UVB bombardment. This occurs in a light-controlled box, which I enclose the inside with a bunch of duct tape, and it's also with a UVB light, which is measured at 100 micromoles of UVB bombardment, which was measured with a power meter. And the samples are placed in a petri dish within the box directly under the light to get kind of even UVB distribution. Control samples were just left on the counter in the environment in which the yeast like grew in. And so for my control counts, I had an average of, for my control counts at 5 minutes of no stress, I had an average pre-cell count of 2.6 million cells and an average post-cell count of 2.1 million cells. Right off the bat here, you can see there's a 19% decrease in cells from pre to post, which is kind of interesting. And then you can see on the graph here, which also communicates these numbers, you have control pre in blue, control post in orange, with a line of best fit. So pre having an R square of 0.0014, post of 0.1394. So there's relatively no linearity here, but that's what would be expected since each cell is an independent value. Wouldn't expect to see some, exponential linear trend occurring. For 5 minutes of UVB stress, the average pre-cell counts were at 3.3 million and post-cell counts at 3.2 million. What's interesting is you can already see that there's a significantly less decrease in cells, only a 3.76 decrease in cells from pre to post compared to the 19% decrease. Again, you can see this in the graph in in 5 minutes of pre in blue, and then 5 minutes of post in orange. Again, with pre of an R square of 0.0101 and post of 0.0591, with low linearity, since they're all independent points.
Hey,
For the
Chloe, you have just
electrophoresis,
about 2 minutes left.
thank you. For the electrophoresis, an initial well was used, causing samples. Oh, sorry. An initial gel was used with a singular well design, causing samples to not stay in their lanes, but successive gels going forward will be run in a multi-well to provide a more comparative analysis. But you can see the two gels, the two protein bands produced over here in this image, which correlated to proteins measuring at 110 kilodaltons and 167 kilodaltons. So, you can see from the data, the cells are not dying due to UVB stress. Control group on average is losing 15.24% more cells than the UVB stress group. This could be for one of two reasons. Either that the UVB stress samples are not dying for one of two possible reasons. One, that UVB stress at 100 micromoles is not significant enough to induce HSPs, so no effect can be seen on the cell counts, or that the UVB stress is significant enough but the cells are not dying since HSPs are being produced to compensate this stress. But electrophoresis need to be run a replicate to further understand this cellular response. For the future, I plan to run electrophoresis in duplicate with 10-well gels, let samples run for longer UVB bombardment times, and let them sit for a slightly longer time period after stress. All in all, preliminary data suggests that UVB stress has little to no effect on yeast cell counts, which could potentially indicate activation of HSPs. Protein electrophoresis was performed to determine HSP expression, and electrophoresis produced data consistent with a higher molecular weight protein like 90, but further testing is needed to require confirmation. If it is possible to selectively induce only HSP90 with UVB bombardment, this has the potential to become an early cancer treatment and glean more insight to the heat shock response. Thank you.